Thank you. So welcome to everybody to this Young Lectures in Life Science. This is actually the last lecture of a series of 3 lectures that are given by Professor Dennis Low. I'd like to give a little bit introduction of this lecture series. And in fact, this lecture series is co-organized by the New Asia College of the Chinese University of Hong Kong and the School of Life Sciences of Chinese University of Hong Kong. And this lecture series are actually made possible by generous donation from the Mr. Yong Memorial Fund. And I'm not sure if you know who is Mr. Yong. He is the one of the founding department chair of the Department of Biology of the New Asia College. And it's a very long history. And this is actually of our honor to have Professor Dennis Low, who is the Li Ka-shing Professor of Medicines of the Chinese University of Hong Kong, as our speaker today. And Professor Low's research is well-leading on the biology and applications of cell-free nucleic acids in plasma. In particular, he discovered the presence of cell-free fetal DNA in maternal plasma in 1997 and has since then been pioneering on the non-invasive prenatal diagnosis using this technology. And this technology has been adopted globally and has created a paradigm shift in prenatal medicines. He has also been making many innovations using circulating nucleic acids for cancer detections, including the screening of early-stage nasopharyngeal cancers. And Professor Low's contribution is internationally recognized. He has been elected as Fellow of the Royal Society, Foreign Associate of the United States National Academy of Sciences, Fellows of the World Academy of Sciences, and founding member of the Academy of Sciences in Hong Kong. And Professor Low has won numerous awards. Just to name a few, for example, he has won the Future Science Prize in Life Science, the Breakthrough Prize in Life Science, and the Royal Medal. Just for your information, the Royal Medal is a prestigious award awarded by the Royal Society of the United Kingdom and was previously awarded to Nobel laureate like Greg Winter, Francis Crick, Fred Sanger, and Max Borus. I understand that there are actually many undergraduate and postgraduate students attending this seminar. I would like to draw your attention, in addition to the great science Professor Low is going to talk about, and you should also pay attention to his magical ability to explain complicated scientific logics in simple terms. And these skills are actually very important in doing science. And without further ado, I'll pass the time to Professor Low, please.
So thank you very much, Professor Wong, for the kind introduction. I'd like to thank the School of Life Science and New Asia College for giving me this honor to give this lecture series. So today, I'd like to talk about some of the biophysical characteristics of circulating DNA. Now, so those of you who have heard my previous 2 talks, I've talked about this relatively new field called plasma DNA-based molecular diagnostics. So in other words, we're now very interested in cell-free DNA, which was swimming outside cells in our blood plasma. And plasma is this yellow fluid in our blood, which represent about 50% of the blood volume. And 2 areas that I've focused a lot of my work on the left-hand side, is the detection of fetal DNA, which has entered the maternal blood during pregnancy. And on the right-hand side is the release of tumor DNA in the bloodstream of cancer patients. So as Professor Wong has said, the presence of fetal DNA in maternal blood was first reported by our group in 1997, in which we used a male DNA from a male baby as a marker. And then we show that this DNA is present from the 7 weeks of pregnancy onwards. And actually by the 10 weeks of pregnancy already reaches some 15% of the concentration. And then by using this technology, we have actually developed a number of prenatal testing, for example, for Down syndrome, which is caused by the baby having extra copy of chromosome 21. So in 2008, we invented this method whereby we basically sequence millions of DNA molecules in blood, and we map them to the chromosome from which it comes from. And then we can plot ratios of different chromosomes in blood. And by using this method, we have a method to screen for Down syndrome with an accuracy of 99.7%. And because the technology is so accurate, it has been rapidly introduced clinically. It's now called NIPT, non-invasive prenatal testing. It's actually done millions of times in over 90 countries. Now, for the last few years, we've been very interested on the biophysical characteristics of circulating DNA. And one of those characteristics is the size of the molecule. So how long is that molecule? Now, for example, if you sequence the length of those molecules, this is what you get. So the x-axis is the length of molecule, and the y-axis is the frequency. So one thing you can say is that the circulating DNA are generally very short molecules. And then interesting, you see actually they have a series of peaks. For example, this is one peak at 166 base pair. And after that, every 10 base pair or so, you have another peak. And then you also find that fetal DNA which is in blue here, is on the left-hand side of this red line, which is mainly from mother's DNA. So in other words, fetal DNA is a little bit shorter than mother's DNA. And then we wonder what are all those peaks? Now, to understand what those peaks are, we have to recall actually that in our nucleus of our cells. So if you map the whole genome, it's about 2 meters long. It's very long DNA. But the DNA actually coiled around some protein molecules called histones. And then it formed this bead-like structure called nucleosomes. And actually, the most interesting bit is this part. So we can see a DNA coiled around the histone and to form nucleosome. And then there's a region called a linker, and then go into the next nucleosome. So this center bit is 146 base pair, and this bit is 20 base pair. And the two together is 166 base pair. So basically what we're proposing is actually this first peak, 166, is basically a core plus the linker. And actually this second bit is just a core without the linker. And interestingly, this is actually one of the fundamental difference between circulating fetal and maternal DNA. In that fetal DNA is somehow lacking in the linker region. And then after that, you will say, well, why do we have all this series of peaks, 10 base pair? And the reason is because the DNA is double helix, and actually it coils around the core histone. And actually every 360-degree turn of double helix is 10 base pair. So this bit could be actually telling us a bit about the secret of how DNA is being cut. Okay, so for the last few years, we wonder, okay, so those DNA molecules are originally very long, and suddenly in blood it's very short. So you must have some mechanism which result in the cutting. And then we think, well, this cutting is likely to be mediated by some enzymes, such as a nuclease. So if that's the case, then one way to tackle this problem is to do a knockout experiment in mice, in which we knock out those nuclease genes in mice. Now, for example, if you take some blood from mice, and then you prepare an Illumina sequencing library from it, then you run a gel, then you should find a band which is about 300 base pair. And the reason is because the band from plasma DNA is 160 plus Illumina adapter, which is about 140 base pair. So you have 300 base pair. And this is in wild type normal mice. So just now we talk about knocking out nuclease genes in mice. So which nuclease to knock out? Now, so you read the literature, then many people assume that this enzyme DNase 1 is a major nuclease in us. So one thing we do is to actually knock out this DNase 1 gene. And of course, what we're hoping is to be able to see that there was some change in the size. But strangely, when I did that, so DNase 1 knockout, there's nothing, no change at all. And actually, we find it so strange that actually we published this negative report in 2018 to say that we don't see any apparent change. Then we wonder, why is that? So maybe in the mice, apart from DNase 1, there might be some other nucleases. So we review the literature, and then we come across this very interesting nucleus called DNase 1-like-3. Now, some literature also call this DNase gamma. And the reason why it's interesting is because this enzyme like to cut chromatin, whereas the DNase 1 like to cut pure DNA. And of course, in the circulating DNA, it's basically chromatin because you have a DNA plus the histones. So DNase 1-alpha seems to be very interesting. So then what we did is that we knocked out the DNase 1-alpha, and lo and behold, apart from this mononucleosomal band, you start to see some bigger molecules. And those are basically of 2 nucleosome size, 3 or 4. And then we also do a double knockout, which basically is the same sort of effect. And then we do some sequencing and plot out a graph. So you can see the x-axis is the size of molecules, and the y-axis is the frequency. And then you can see the wild types in black, and DNase 1-alpha knockout is in red. So once again, you can see those long molecules. Now we have this preliminary data suggests that DNase 1-like-3 will cut on the circulating DNA. But then we wonder, how about the bits that it cuts? Does it have any preference? So we decided to take a look at the sequence at the end of those molecules. We call this end motif. So we decided to take a look at 4 base. So of course, if there's 4 base, because you have ACTG, then theoretically, that would be 256 possibilities, so 4 to the power 4. Now, if the whole thing is random, basically each of those motifs should be present at a ratio of 1/256. So is that the case? So when we did the actual experiment, we find that in wild-type mice, the top motifs actually is present at a ratio much higher than 0.39. So you can see 1.5, 1.45, etc. And then when we knock out the DNase 1-like-3 gene, we find there's a significant change in those ratios. Now, if you're very observant, you'll find that the top 6 motifs all actually start with CC. So it suggests to us that actually that the DNase 1-like-3 might like to cut CC molecules. So then we decide to actually look at those 6 together. And we find that in wild-type mice, plasma DNA with the CC motif at the end represents 7.5% of all the circulating DNA molecules, which is quite a lot. And if you knock out DNase 1 gene, nothing happens. If you knock out DNase 1-like-3, then the motif frequency becomes half. And you do a double knockout, it's still the same. And then we decide to explore this a little bit more detail. Now, because pregnancy is quite interesting, because you have a fetal and maternal DNA in the circulation, I think pregnant mice would be an interesting model. So then what we decide to do is that we have some female mouse, which is black mouse, okay, black 6, and then we knock out the DNase 1-like-3 gene. And then on the male, we have some white mice, and then this is basically wild type. And then we make them. So theoretically, the baby should be heterozygous. So 1 copy of DNase 1-like-3 gene and 1 copy which is gone. And then we like to see what happens. And of course, we wonder, well, in mice, whether the fetal mice, like the babies in the pregnancy, would also release the DNA into circulation of the mother. Now, just remember some of the statistics. Now, if it's non-pregnant, if it's wild type, then 7.5% of circulating DNA have the CC. But if you knock them out, then this motif frequency is half. But now in pregnant women, in pregnant mice, if it is knock out, then actually what we find is that the motif frequency is similar to non-pregnant. And if it's wild type, it's also similar to non-pregnant. Now the interesting bit is if the baby and the mother have different genotype. So remember, if the mother have the knockout, but the baby is heterozygous, then you can see there's a partial salvaging of the frequency. And finally, if you just look at the fetal DNA specifically in the mother's blood, because we know that the baby has got this father's DNA, which is from these white mice, we can specifically look at those DNA. They can see this further salvaging of that motif frequency. Okay, so now basically the conclusion of that is very interesting. So in other words, in this pregnancy model, we know there are two things which happens. The baby will release its DNA in the mother's blood, and also having the baby alone, it will already correct the motif frequency of mother partially. So suggesting that the baby will have one copy of the gene, so the baby will produce an enzyme, and the enzyme will go into the mother's blood, which will then help to salvage the mother motif frequency. And also secondly, because the baby is the origin of the enzyme, so the DNA released by the baby will be even more salvaged. So those are the two conclusions we get from this. So this work we think is interesting because it's the first link of nuclease biology and circulating nucleic acid. And it suggests that this sort of phenomenon is associated with some physiology like pregnancy and pathology, such as when you knock out one copy of a gene. But of course, the data I showed you just now is generated in mice. The question is, what happens in human beings? Now, so in collaboration with a group in Toronto and a group in Italy, we're able to actually identify very rare individuals who have got mutations of a DNase 1-like-3 gene, human subjects. And interestingly, those people have familial systemic lupus erythematosus and SLE. So there's a type of familial autoimmune disease. And of course, as all of you will aware, one of the hallmarks of SLE is that they have anti-DNA antibody. So it's interesting. So maybe because they cannot clear those DNA so well, that somehow they're predisposed to producing anti-DNA antibody and the concomitant pathological effects. So basically what happened is that in this human cohort, we sequence the normal individual, and then we basically have the very typical picture we see in the plasma DNA size. We see this peak at 166 base pair. And then we look at the parents of the patients. Now, because this familial SLE is an autosomal recessive condition, so it means that the affected offspring will have 2 copies of mutant gene, but whereas the parents will only have 1 copy each. So when we look at the parents, basically the profile is the same as control. So now it seems that you only need 1 copy of this functional DNase 1-alpha gene to have a normal profile. But now when you look at the individual with homozygous mutation, then you basically see there is a change. There's an increased amount of the long DNA. And also you can see that in the very short molecules, there's also some increase. And this effect we have seen previously in SLE, and we have shown in other work that this is due to the anti-DNA antibody effect. Anyway, if you look at the longer size, it's basically replicate what we find in the mice. And this is a heat map to actually show the motif frequency. And then we have a healthy individual, sporadic SLE, so unrelated to this mutation, and then some control individual with other conditions, like people who have transplantation and people who have arthritis, and the parents of those patients. And this on the right-hand side is the people with homozygous mutations of DNase 1-like-3 gene. So I think one thing you can see immediately is that basically there's a lot of change in the motif frequency. So you can see that in normal individual, it's mainly CC, but however, in the people with homozygous mutation, then the frequency of CC drops because the blue means decrease. So once again, this replicates what we find in mice. And then when we actually look at the motif frequency of those CC motif, we can find a wild-type individual is about 10% CC. So as you remember, in mice, it's about 7.5%. But now in the mutant subjects, there's about half to become 5%, which once again is very similar to what we get in the mice. So in other words, this DNase 1-like-3 biology and the motif technology have implications to human subjects. But then the question is, can we use this motif technology as a new type of diagnostic marker? So we decided to take a look at cancer. Now, interesting, when we actually look at the gene expression profile of DNase 1-like-3 in different tumors, we just look at the data, for example, in TCGA database, we find that in most cancer types we look at, there's a reduction in gene expression in DNase 1 like 3 in the tumor. For example, this data here is in liver cancer subject. You can see in tumor tissues, there is a reduction in this level. So now if there's a reduction in DNase 1 like 3 in cancer, then theoretically, there should be a reduction in the CC motif in the plasma of cancer patient. And indeed, we find that is the case. So this liver cancer patient, and this is the reduction of CC motif as compared with hepatitis B carrier and control subjects. And then we find this to be true for other types of cancer. So it seems to be a very interesting general way you can actually detect, monitor, maybe screen for cancer. So that's DNase 1 like 3. And then we think, well, since we have now got all this technology motif and this knockout model to establish DNase 1 like 3 to be a key player in fragmentation plasma DNA. So maybe it's time to actually try to apply some of this too back into DNase 1, which was the first enzyme we look at. And also maybe we can actually look at other nucleases. And one very important nuclease is caspase activated DNase. And some people also call it DFFB, which is DNA fragmentation factor beta, which is related to apoptosis. So basically we have an experimental setup, which is very similar to before. So we basically knock out each of those nucleases. And then we, in addition, we deliberately take some blood from mice and actually leave them room temperature to actually allow the new plasma DNA to be released in the blood. Because from our previous work, we know that if you leave blood in room temperature, there's a gradual increase in the plasma DNA concentration. So we'd like to see whether knocking out those nucleases will do anything to the profile of the newly released DNA. Now this time, because the complexity, we have 3 enzymes to play with, we decided instead of looking at 4 motifs, which is 256 possibility, we just be simple. We just take a look at the N single motif. So now we just split the plasma DNA in 4 types, those with N's, A, C, T, and G, and then see what happens. Now, for example, if you take some wild type mice, and then you fresh, and then you sequence it, and those are the distribution of the concentration of plasma DNA N with A, C, T, and G, so on here. But then when you leave them in room temperature for 6 hours, you can see that the motif frequency changes. Then you have some newly released DNA, which is predominant A, which is green, and G, which is yellow. So now what we did is that we knocked out the SFB gene, the one involved in apoptosis. And interestingly, you can see that at time 0 is the same, but however, at 6 hours, then all this increase in A and G is gone. So now it's just from this work, you know that basically the FFB is responsible for cutting DNA N in A and G. So basically we have this conclusion, and we can plot that it's A more than G and more than other things. And you do the same experiment, and you know DNase 1 like 3 is C bigger than T, etc. And do the same thing for DNase 1, and then you know it likes to cut T. And of course, in the future, you can use a similar approach to tackle other nucleases. So after we've done this bit by bit, we'll basically build up a model about how it's fragmented. And this work was published in 2020 in the American Journal of Human Genetics. Okay, so up to now, we've talked about this circulating DNA. But of course, one question is that we wonder whether what I've drawn here, which is a double-stranded DNA all along, is that correct? I mean, is it possible that you might have some circulating DNA which is partially single-stranded? Now, for example, maybe at the end, you have a bit which is protruding. So we call this a jagged end. But of course, if you want to answer that question, you have to develop a technology to allow you to assay the jagged ends. So we think one way you can do that is to copy it, to repair it. And during the repairing process, you can add some sort of marker which will allow you to detect it. Now, for example, in the repairing process, I can have unmethylated C. And then after that, I can detect the unmethylated C versus methylated C by bisulfite sequencing. So bisulfite is basically a process that when you treat DNA with sodium bisulfite, and unmethylated C will be changed into U, whereas a methylated C will remain a C. And if you look across the genome, if you look at CG sites, about 75% all CG site across our genome is methylated. So in other words, if I add in unmethylated C during the repair process, I should see a reduction in methylation density if there's jacket ends. And indeed, this is what we find. So we discover the present jacket ends in plasmid DNA. And then this we call the jacket index. But as a control, you just extract some nuclear DNA and you fractionate it, break it up by sonication, then you find that the jacketness is very low. And then by using a similar approach, we can also ask how long is the jacket? And then which we find that 12% DNA is blunt. So then 12% DNA is just completely double-stranded, but the rest are all jacket. And they could range in jacketness from 1 nucleotide to over 70 nucleotides. So it can be a whole range. And furthermore, we find that fetal DNA have more jacketness compared with mother's DNA. And the jacketness is focused on DNA, particularly between 130 and 160 base pair. So probably once again related to nucleosomal structures. And actually, because fetal DNA is more jacket, so we can actually use the jacket index to deduce how much fetal DNA I have in a particular sample. And this is important because the fetal DNA concentration in a pregnant mother sample is the #1 determinant of the accuracy of non-invasive prenatal testing. And then we wonder what is causing jacketness. So we think it's likely to be once again related to the nucleases. So what we did is once again explore this by the nuclease knockout mouse model. Now, for example, look at the wild type mouse. This is the jacketness. And if I knock out the DNase 1, there's a reduction in jacketness. If I knock out DNase 1 like 3, there's an increase in jacketness. So basically what our working model now at the moment is basically when DNase 1 like 3 cuts, it's a tendency to just cut on one of the strands, leaving the other one. So in other words, it's a cause of jacketness. So if you knock it out, then they take away this effect. But on the other hand, DNase 1 like 3, it like to cut both strands. So in other words, it like to create non-jacket DNA. So if you knock out DNase 1 like 3, then you leave the effect of DNase 1, so that will increase jacketness that way. So I think this jacketness is interesting. This is an intrinsic and newly discovered property of plasmid DNA. And the fetal DNA have more jacketness than the background maternal DNA. And recently, we've also shown that cancer DNA is more jacket than non-cancer DNA. And once again, it's related to nuclease activities. And this work was also just recently published. Okay, so up to now, I've talked about various ways in which our genome is fragmented and then released into plasma. And this field now actually has been called fragmentomics. Actually, when you look at literature, the first use of the term fragmentomics is actually in the 70s when people talk about fragmentation of proteins. But of course, now we can similarly use it to describe DNA. So now let's take a look at the other side. So we talk about some small fragments, but on the other side is how long can they be? So how intact can they be? Now, because up to now, I've shown you about this graph. This is actually a graph that we have plotted in our 2010 publication in Science Translational Medicine. But at that time, we have actually used the Illumina as a DNA sequencer. And actually, since then, most people who work in this area will use Illumina sequencer. And one question we ask is, well, the fact that we don't see this long DNA, longer DNA than this, could it be because of Illumina sequencer? So what happens if we use a single molecule sequencer like that of Pacific Bioscience, PacBio, which use single molecule real-time sequencing? Is it possible that I might see longer circulating DNA? Now, for example, as I showed you just now, you use Illumina sequencer, this is what you get, 200 base pair, and you may get a little bit more. But interesting, recently, when we find the PacBio sequencing, we find in circulating DNA is a whole population of long molecules. which were not previously discovered. Actually, when you look at first trimester, second, and third, you all see those long molecules. And actually, the amount of long molecules increase as gestational age increases. And actually, those are quite substantial. When you look at the median value, you find that it ranges from 15 to over 30% of DNA are long. And those have not been discovered before. But then you say, well, what is the significance of this? Now, what is the significance of this? If you look at a single nucleotide polymorphism in our genome, on average, we have one SNP every 1,000 base pair. And of course, if your plasma DNA is 160 base pair, then you need several plasma DNA molecules before you have a chance of covering one SNP. But now, if I have long plasma DNA molecule, then theoretically, I should have much more increased chance of seeing those SNPs, the differences between baby and mother. And indeed, we find that now with this long plasma DNA molecule, we can find a fetal DNA molecule in mother's blood, which is 16 kilobase long. And just one molecule, I can decipher the haplotype, the combination of different SNPs. So this baby I know is a CA, CT, CT. Those are SNPs which are separated by could be a 1,000 base pair. Whereas in the old days, when I used Illumina sequencing, I need multiple reads, many reads, before I can even have a chance of seeing that. And even then, I have difficulty in connecting the SNPs, in working out the haplotype. And the mother side, even longer, can have a 24 kilobase molecule in the circulation. And one molecule, I can work out a lot of haplotype. And also because it's so long, I can also work out where it is coming from. And one of the ways we can work out where it's coming from is by looking at DNA methylation profile. The reason is because within our body, different cells have different methylation profiles. And then we can actually use this approach to deduce where they come from. Maybe even do it on one molecule because it's so long. But then, of course, we have a problem. One problem is that, as I mentioned just now, you will try to detect methylation. You can use this method called bisulfite conversion. But for those of you who have played with bisulfite before, you know this is a very nasty process. This chemical will destroy sometimes 90% of the DNA you put in. And furthermore, they may actually make the DNA to be shorter. So it's not very good because we like to look at long DNA. So I was wondering, is it possible that I could do a DNA methylation analysis without using bisulfite? So we think maybe that with this single molecule sequencing from PacBio, is it possible that during the sequencing, the methylation profile will actually change some of the kinetics of DNA sequencing? Now, for example, here we have one DNA molecule, and then we have a CG, and I want to measure the C methylation profile. A methylated C is in black, unmethylated is in white. And then I add in some sequencing adapter in the end, and then I can sequence. So you can see in Pacific Bioscience sequencing, I can sequence both strands of DNA molecules, the Watson strand and Crick strand. Now, if you look in the literature, you'll find that Pacific Bioscience have actually tried to do this before, a few years ago. But they say that the accuracy is like 5%, which is very bad, right? And so we're wondering, is it possible that they might have some hidden information in there? Now, so this is basically what happened in the Pacific Bioscience sequencing. So DNA molecules and the polymerase come and copy the DNA molecule, and they incorporate A, C, T, and G, and each one will give some fluorescent signal. So give a T, the green signal, and G, etc. And then we can actually measure the time duration between different signals. And then we can also measure the duration of signal itself. So duration of signal itself is the pulse width, how long that pulse is. And then the duration between signal is the interpulse duration, IPD. So we developed this thing, we call it holistic kinetic model, HK model. So in other words, we look at different kinetic parameters of the DNA polymerization. And then let's say I want to measure this methylation state of this C, and then I create a measurement window across the C. and then we measure the IPD and PW for all these things. It's almost like you're driving a car, and then in front there is something that you run into, that you hit a box, let's say in the middle of the road. Before you hit, you're probably already doing something, right? You're doing some correction maneuver, you're pressing a brake, etc. And then you hit the box, and then your car suddenly will stutter and then change some more. So in other words, what we're saying is that the fact that this C is methylated or not, either before or after the polymerase has copied the C, it's probably going to change in the kinetics. So you do that in the Watson strand, and you also do that in the Crick strand, and then combine the two tables. So now when you look at this table, it looks like a two-dimensional thing. It's almost like a picture. So what we're asking, is it possible that we can use artificial intelligence? There are many different ways we can actually handle pictures, like use a convolutional neural network, CNN, to process picture. Is it possible within this picture of this polymerization, there might be some hidden signal of the methylation status that we want to look at? So we do a training set, and then we use a validation set. Now to train this, and of course you need to have some gold standard. So what we did is that we take some DNA and then add an enzyme which will methylate all the C. So this is the positive control. And then we have negative control. So we start with some genomic DNA, and we use PCR to amplify DNA. And of course the PCR, they won't create the methylation. So this is a negative control. And then when we do that, we actually find that we can differentiate the positive control from negative control very well. And we use different versions of the bacterial bioscience sequencing reagents, and they all work. And then we measure this area under curve. So this is a receiver operating characteristic curve, ROC curve. So if your test is rubbish, it'll be along this horizontal line. If it's perfect, you'll go all the way to the top left-hand corner. So, and then you can measure how good it is by area under curve. If it is 1, that is perfect. So you can see that this test is pretty good. So area under curve is between 0.94 to 0.97. And this is the training set. And this is the validation set, which once again confirm the data. And then after that, we decided to test on some complex DNA samples. Now, for example, we test on placenta. We test on some liver cancer tissues. Some normal liver tissues, non-cancer. We test on blood cells and some cell line. And then we use bisulfite sequencing on x-axis as a gold standard, and the y-axis is our HK model. You can see that basically correlate extremely well. The correlation is 0.99. Actually, when we delve into individual genomic region, say within chromosome 1, 2, 3, 4, 5, etc., and the inner ring is your HK model, and the outer ring is the bisulfite sequencing, gold standard. You can see it fits extremely well. So all the colors matches in different genomic region. And for each of those, you can actually plot a graph, which actually once again show extremely good correlation, as shown in the bottom here. So we think this is a very important development because this direct methylation readout is very direct and very intuitive. Now, for example, if you use the old days bisulfite sequencing on Illumina, you have a series of very short reads, and then you can see a little bit of readout here, there, but you cannot connect the whole things. But on top here, when you use the HK model, you have one read which go all the way from the black region all the way to the white region. And furthermore, let's say in the top here is a reference human genome, and in the middle is HK model readout, the bottom bisulfite sequencing. Now, when you do bisulfite sequencing, when you say you see a T, you actually never know before you look at the reference human genome. You don't know whether this T is from a reference human genome or whether the reference genome is a C, which is unmethylated, which then got converted into a T. So you don't know that. But however, with HK model, I can just read. I see this is a C, which is unmethylated. So much more direct and intuitive. So this work was just published early last year. So now we wonder, okay, can I use this HK model directly on plasma DNA? If I do that, then I have the DNA sequence in plasma, and also have a methylation status. And indeed, and then I can just map the methylation patterns of this circulating DNA directly with where it comes from. And indeed, we find that we can do that. And also performance is very good because by using this approach, I can have long plasmid DNA. The longer it is, the more CpG site it has got. And you can see here, for example, the red line is fragments bigger than 600 base pair, and the black line is short fragments. You can see that by having longer fragments, I have a better performance in this RC curve. So the 0.91 is bigger than 0.74. Of course, now, if this model is so powerful, so you can know the sequence, you know where it could come from, then I can actually probably use it to diagnose a variety of genetic diseases. So we decided to test it out. Now, for example, you can imagine in a pregnancy, let's say the father has got 2 chromosomes, and on top is one of chromosome, bottom another, and there's a gene here, and the mother similarly with 2 chromosomes. And then by sequencing a mother's plasma, I can actually work out individually where a particular DNA molecule is coming from. And then by looking at the mother, at the DNA molecules in the mother's blood, I can find those with a methylation profile which fits that of a placenta, which means it's coming from a baby. And then I basically know what sort of polymorphism the baby has inherited. Now we decided to test this in a family in which is at risk of having Fragile X syndrome. So now it's a mother is a carrier of a pre-mutation of Fragile X. So Fragile X is a genetic disorder which affects mental retardation. And then they have a previous baby who is affected Fragile X. So the mother's pregnant again, and she wants to know the risk of this new baby. So you want to know whether her at-risk allele has been passed on to the new baby. So when we look at the mother, we find the mother has 2 X chromosomes, and one is a blue one, and the other one is a yellow one. And the blue one is the one which carries the pre-mutation. And we know the blue and red because we know the polymorphisms. And then by looking in the mother plasma DNA, and by using the HK model, I can basically work out the baby's X chromosome. I know the baby has inherited the yellow one here, the blue one here, and the yellow one there. And so lo and behold, I know basically the baby has inherited the normal bits, the normal Fragile X gene from mother. So in ours, it's not a risk. So you can see that by using this technology, I can very easily work out genetic recombination or meiotic recombination. Now, the previous gold standard of this sort of approach was published by our group in 2010 in Science Translational Medicine, in which if you try to detect as fetal genetic disease, I will need a lot of reads, like 4 billion reads. So of course, that's very expensive. So for example, you can see the previous technology is this blue technology. I need many, many reads before I can reach a high diagnostic accuracy. But now, because I have such a long plasmid DNA molecule, I read very little read, is already close to 100%. So I think this is going to basically change the provision of a single gene prenatal diagnosis. Apart from that, you can also test in some other conditions. And one condition is called Preeclampsia, which in Chinese is called 妊娠毒血症. So which is a condition in which a pregnant woman has high blood pressure, urine, protein in urine, and globally it's about affect 5% of pregnant women. So because of this importance, so many people have been trying to develop markers for preeclampsia. And interesting, we actually find by using this long DNA technology, I find that actually women with preeclampsia have a change in the amount of long DNA, have a reduction in the amount of long DNA, just like that. And for example, I see this normal pregnant woman, this is preeclamptic pregnant woman, this reduction. And this is using Pacific Bioscience sequencing. But however, previously people don't know that because if you sequence by Illumina, you don't see any of that because you don't see the long molecules at all. So anyways, this work was just recently published in Christmas last year. And we think it's important because up to now, in the 20 years of non-invasive prenatal testing, we are based on short DNA molecules in plasma. It's like I'm sending you a text message. Each message can only be, say, 50 words. But now, for the first time, I can now send you a Word document, which is 2 megabytes long. So I think this is it's going to really change the field. So we're very excited by this latest development of long cell-free DNA diagnostics and direct methylation analysis. This allows you basically to see details which you cannot see before. So I think in the coming years, we should have many unrealized research and diagnostic potentials. So finally, in summary, hope I've convinced you that plasma nucleic acid is really a treasure trove for molecular diagnostics. With it, the era of NIPT is already with us. And today we talk about an emerging field called fragmentomics. We talk about fragment size, end motifs, and jacket ends. We also talk about this intriguing link to nucleus biology, and also about this new development of long cell-free DNA and direct methylation analysis. And actually recently we've summarized our thought in this review in Science. So if you're interested, you can look this up. And finally, I'd like to thank individuals from my group for generating data which I present to you today. So thank you very much for your time.
Okay, thanks Professor Lo for his excellent talk. And now we go into the Q&A section. Okay, so if you want to raise a question, either you can type it in the Q&A box, or you can raise your hand to see if I can ask my technicians to switch on the mic. Okay, I already see one question from the Q&A box. Okay, let me read it out. Is it possible that the plasma DNA actually coming from apoptosis? And do these plasma DNA have a preference from specific genomic origin or randomly? And he also asked whether how does the plasma DNA methylation status maybe using ONT-DDS as a more powerful indirect? Okay, Professor Lo.
Yeah, so it's a very good question. So I think that the plasma DNA basically is released by cell death, and then different ways of cell death. So apoptosis is one way, and necrosis is another way. So depending on the type of cell death, then you see characteristic that way. So another question is about the genomic region. So actually the genomic region will affect the biophysical characteristics of the plasma DNA, which is released that way. Now, for example, just now I told you that the nucleosome structure will affect the structure of plasma DNA. Then of course you then think, well, what happened if it's released from a nucleosome-free region, like the open chromatin region? Then yes, the characteristic will be different. And similarly, if you have something which doesn't have histones, like mitochondrial DNA, then it's changed as well. For example, if it's from mitochondrial DNA, some of them are circular, just like mitochondrial DNA itself. And also if the stuff is released from bacteria, and bacteria doesn't have histones, and the structure is also different. So I think it's a lot of things for us to work out.
Okay, thank you. Any other questions from the audience? I've got one more. Okay, I would like to know more about how could we knock down the enzymes, DNA in the mouse in the experiment. Okay, yeah, Professor.
So you have different ways. So nowadays you can probably use CRISPR. You can probably, in the old days, you can use homologous recombination. And then, of course, you can also use gene knockdown, RNA interference.
Okay, thank you. Okay, actually, I myself has a question, Professor Lo. We know in one of your early slides, I see that when you draw the way how it cut the chromatin, I see the arrow actually targeting the minor groove of DNA, or is it? Did you know anything about this, or it just so happened you just draw it?
It's minor groove, yeah.
Oh, really? Okay, so that's scientifically correct. Okay, so
yeah, so we actually look at that from the. I mean, actually, DNase 1 has been crystallized, so that is from that. And actually, even last week, actually somebody uploaded a preprint on DNase 1 like 3 structure. So I think we look at that very carefully.
Okay. Because when you look at the consensus sequence, it basically is a CC, but the chromatin is double-stranded. So you know what I mean. So CC the other way it will be a GG, right? So I guess is it known that how it recognized, or it will be known in a short period of time because of the preprint?
Well, I think that people probably has not looked at that sufficiently in detail, and this is what we're going to look at.
I see.
Yeah.
I see. Thank you. Okay, so okay, there are just a few more questions in the Q&A box. Okay, the next one is, according to my understanding to apoptosis, there will be DNA fragmentation. Will this affect the analysis of the DNA?
Yeah. So basically, what happens is that all of the processes, let's say that you have this first fragmentation of DNA, that is the DFFP, basically, that step. And after that, there will be the 1L3 and then DNase 1. So all of this will make the mark on your plasma DNA. So now basically, when we use the Pacific Bioscience, we sequence very long DNA. We can see that from the very long, let's say 20 kilobase, down 10 kilobase, 5 kilobase, etc. All the long guys, all the DNA molecules of different size, they have their own particular structure. So when you're long end, you actually see the DFFP signature. And then in the mid end, you see a 1L3, in the very short end, you have the DNase 1. And I'm sure there will be other guys that I don't know yet, which I'm going to find out.
Okay, great. Okay, I also see actually one question from my student. Actually, will be your student in the future. Okay, she actually asked if it's possible to detect the jagged end at the 5' end. I think in your example, you have the 5', one of the. Can you do the other way around?
That's very clever. So maybe we need her to help us do that. So we're constantly working out different ways in which we can play this game. So I mean, theoretically, there are different ways you can play it. And one way is to put an adapter. So let's say I have this jagged end, I can put in an adapter. But then, of course, it needs to be very tedious because I don't know how long the jagged end is, right? So if there's 2 or 5, then I need to have an adapter of that particular size. So I might need a whole range of adapters.
Yeah, that's interesting. Okay, I've got questions. Okay, okay, from Alfred actually. His question is for fragmentomics to be applied in cancer diagnosis. The tumor seems to beneath the nucleus. Okay, nucleus 1 or nucleus 1L3 expression in a aberrant rate. So is it correct? If so, is there any way to improve the cancer diagnosis?
So interestingly, when I look at the TCGA, the gene expression profile of tumors, I would say easily 90% of those tumor different types have reduction DNase 1 like 3 expression. And then also some of them, and actually many of them, have increase in DNase 1 production. And actually DNase 1 production, I've actually seen the literature has something to do with prognosis as well. The increase is bad prognosis. So what this suggests to me is basically this technology will allow us to detect and probably prognosticate people. Yeah.
Okay. Oh, now I also have another question. So, the question is, I've seen that you have utilized a lot of PacBio third-generation sequencing for your research, at least for what, and I have also heard that the third-generation sequencing is prone to many more sequencing errors than the second-generation sequencing techniques. So may I ask that if that was an issue to your research, or did that somehow affect the reliability of the finding, or once giving you a challenge to your research analysis?
So thank you for that question. So basically, the currently widely available third-generation sequencing are 2 types. One is PacBio, one is Oxford Nanopore.
Yes.
Actually, the students' comment is more relevant to Oxford Nanopore. So Oxford Nanopore currently has a sequencing accuracy around 94, 95%. And whereas the Illumina will be 99.9 something. And PacBio is even high, 99.999. And the reason is because of PacBio, as you remember just now, it makes a circle. And what it means is that the polymerase can go around the circle many times. I mean, you can even go around 30 times or even 100 times. And then the sequencing result is the consensus of that. It's called circular consensus sequencing. So PacBio is extremely accurate. But whereas the Oxford Nanopore, of course, is just go through, swim through, and that's it. So that is more applicable to that one. But of course, there are always pros and cons because the PacBio only currently have 8 million wells. and you can only do it on 4 million sequences. But whereas a NovaSeq from Illumina can do 10 billion. And then nanopore, some can do 3,000. And yeah, so that is, yeah, you have to balance the pros and cons.
Okay, great. Okay, do we have any more questions from the audience? Actually, I do have another one, Professor Low. You know, when you do when you try to measure the jacket index of the DNA, do you need to, you know, methylate all the DNA in the sample beforehand so that you can have a clear cut what is not, you know, what is extended or not?
You don't have to. The reason is because the bit which is double strand, I know is 80% already.
Oh, I see.
Yeah, we're born that way. So that is our structure. And I'm only looking at this bit.
Okay, so which means that because the sticky, the extended end are so short, by chance it wouldn't have, any ambiguity in the methylation. Thank you.
Yeah. Okay. Actually, maybe I'll say one more thing. So this is one way, and then of course we have all sort of enhancement. And that enhancement is to look at the ends such that they are 2C, CC, and then we look at the. So CC normally the C is not be methylated. And I sort of look at one guy is methylated and the next guy is unmethylated, or vice versa. So that is the junction of the jacket ends.
Yeah, that's interesting. You know, the other thing. Sorry, now I've been keeping all the questions. It's interesting. So because you mentioned that it only cuts, apparently it's not only it preferentially cut at the minor grooves, but you think about the DNA wrapped around the histone, right? So even you have a CC, it so happened the CC is sort of buried, in the cycle. Not so happen it's exposed, in the minor groove that are accessible to the enzyme. Then it will not be cut. Am I right? Or
yeah, I think that we need to look at the crystal structure. But because the reason why I'm hesitant is because there are many facts in that. Not the least that if you look at the nucleosome structure, interestingly, when you look at nucleosome, there is a periodicity of the frequency of those dinucleotides. So it's a lot of effect we have to play with.
Okay, I've got another question from Alfred. Okay, there are different ways of cell death, like apoptosis and etc. Do you think that the proximal nucleic acid characteristic may deduce what types of cell death that in sample?
Yes, I believe so. Yes, I think now we have the tools for you to play with all that stuff.
I guess that this is mainly due to apoptosis, from what I know from your research, right?
Exactly. Of course, I think that would be interesting if you look at particular. You can have some mouse model in which, for example, you can give some chemicals to induce necrosis. For example, people use paracetamol to induce liver cell necrosis, carbon tetrachloride, etc. So I think that would be a very interesting thing to look at.
Okay. Okay, I have got one. I still remember when Professor Low. I still impressed me. You make an analogy between tumor and pregnancy, right? And does it mean that I'm not a does it mean that the fact that when you have a pregnancy you've got a lot of DNA from the fetal DNA, is it because you've got a lot of apoptosis?
Yeah, because basically the cells, any cell which grow a lot, then have a high chance that they also have some cell which dies. So if you look at placenta, they have particular apoptotic frequency, let's say 1% of cells. And then you calculate from how big the placenta is, and then you can calculate how much DNA you are expected to see.
I see. That's interesting. That's interesting. Okay. So okay. Any questions? I guess okay. So if not, okay. Before you go, okay. Kenneth, can you show up the slide? So before you leave the, first of all, thank you very much for attending, Professor Low's excellent lecture. And before you go, I'd like to attendants to, fill up this questionnaire of the lecture series, and then you can use this QR code and then scan it, and then try to fill up the questionnaire before you leave. And finally, I would like to thank Professor Lo again, okay, for his excellent talk, and that will end the final lecture of this Yung's lecture series. Thank you very much. Thanks for your attention.
Thank you.
Thanks, Dennis.
Thank you.
Okay, bye-bye.